So tonight we're very lucky to have a team that's accomplished some amazing things that combine CNPS and the Laguna. Betty Young, who organized this, is a board member. She's a former president. She's a horticulturist with a degree from Davis. She's a fellow of CNPS. I don't know how many years ago you were nominated by Liz, but anyway, you're a fellow. And it was Betty that wrote the grant for the shade house and the expanded nursery that we share with the Laguna Foundation. We're very lucky. There are thousands of hours people spent growing plants at their homes, and now everything is centralized and well organized. It's really impressive. And we don't have to pay rent for the Veterans Auditorium, which is marvelous. But anyway, I'm going to have Betty introduce Asa Voigt and Ayla, who are her compatriots in this big project. And I'll let you take it from there.
Okie dokie. So I'm introducing our two other speakers because I work with them constantly at the nursery, and I've gotten to know and respect them both. So if you haven't read the newsletter or don't know why you're here tonight, we're talking about both organizations' work on endangered plants that are truly on the brink right here in Sonoma County. We always hear about endangered species away in the Amazon and you think, oh, I hope they're doing something. Well, we have the same problem right here. And so we're going to talk about what many of us in this room have been doing over the last almost actually 50 years. And so our first speaker is Ayla Mills, who is no longer my cohort, but Lynette Bauer's cohort in managing the Laguna / CNPS nursery. I've known her from previous jobs, and she has been a wonderful nursery manager. She's a great propagator, and I do not say that lightly. And she had her master's degree from Chico State, and many years worked as nursery manager at Sonoma Ecology Center before she came to Laguna Foundation. And worked on several endangered, including the Kenwood checker balloon, endangered species there. After anyway, CNPS helped with that too many years until SEC took over pretty much. And she's worked on many other species as part of Laguna Foundation, who we think of as our partners because of the nursery, and I think that relationship has grown into a great relationship between her and Asa, leading up work at Cunningham Marsh, along with our volunteers. So we're going to each be doing a presentation on the different species or group of species, and Ayla will be up first. There you are. Good idea.
Nice. So do I have to hold this microphone the whole time?
Oh, do we? Do that? Yeah, we gotta.
Oh yes, I think I prefer that. So you can't see me shaking that way. So this just clips on.
Yeah.
This.
I'll turn this one on.
Okay.
Put it right at the B. No.
Down here.
Turning that one. I'll turn this one off first, and then I'll turn that one off.
Right.
Okay.
Oh, thank you. You can put it.
Okay, that's better. Well, thank you so much for having me. And it is just an honor to be introduced by Betty Young. whenever I got the opportunity to work at the Laguna Foundation, one of the main drivers for me, to be honest, was to get to work alongside Betty. She's like my idol. And so to get to give this talk with her is really just a blessing. So thank you so much for organizing all this, and alongside Asa as well, who I also very much enjoy working with. Yeah, is this sound echoey?
Turn it down.
Oh, is it not over here? We're getting it worked out, you guys. We'll get there. So yes, so today I'm going to be talking to you about the endangered plant work that I've been doing whenever I was at Sonoma Ecology Center, and now with the Laguna de Santa Rosa Foundation.
Is that better?
Is that better? So, I could just talk. I'm pretty loud.
You gotta talk for her to adjust.
Okay, all right. Is it working? Is this better? Doing good? Okay, let's get started. So today I'm going to be talking to you about the work that I've been doing to amplify seeds of endangered plants. And I'm going to tell you a little bit about the endangered plants that we're going to be talking about. I'm going to tell you about their habitat requirements and some of the threats to the endangered plants. We'll talk about the plant propagation methods that I used to grow them. And then I'll give you some exciting results at the end, and we'll acknowledge a few people. And if we have time, which I doubt we will, there is a video at the end. All right, so what is seed amplification? And that is the process of growing wild collected seeds to increase the amount of seeds that you have. And so that's what I've been up to with all of these endangered plant projects, is taking a small amount of wild collected seed and then growing it in the nursery or in our hoop house to produce more seeds. So that's what this presentation is all about. And these are the beautiful plants that I have been getting to grow. And the first one was the Kenwood Marsh Checker Bloom, then was Sonoma Sunshine, which I have to say is one of my favorites. And then came Burke's Goldfields, and then Sebastopol Meadow Foam. And these are all endangered, state and federally endangered plants. The first one that I got to grow was the Kenwood Marsh Checker Bloom, and this was when I was at the Sonoma Ecology Center. And the Kenwood Marsh Checker Bloom grows in freshwater marshes. So all of these plants are also wetland species that I'm going to be talking about today. There's only 2 populations left in the world of the Kenwood Marsh Checker Bloom, Sedelcia oregana, subspecies Velida. And one of them is at the Knights Valley property, and it hasn't been surveyed for years because the property owners don't really want anything to do with people trying to manage the endangered plants at their site. But the other one is at a vineyard, actually, the Deerfield vineyard, and that landowner has been very cooperative and is in a 10-year agreement with NRCS and U.S. Fish and Wildlife Service to help manage the Kenwood Marsh Checker Bloom on their property. So, like most wetlands, a lot of these species, are suffering due to human involvement in altered hydrology, habitat fragmentation, habitat loss, competition with invasive plants. And because of all of these things, the loss of their genetic diversity and the numbers that they need to keep reproducing. So here's a picture of, the Kenwood Marsh Checker Bloom, what it's up against. So it's pretty much surrounded by non-native blackberry. And that's one of the things that CNPS has been helping do with this plant over the last, I'm not sure how many years. We'll save questions till the end. Is helping control the invasive blackberry on the property. But my job was to take this seed that they had collected from the wild population back in 2007 and 2019 and see if the seed was still viable. And then also try to produce more seed. So that's what I did. And there was no real information out there how to grow the Kenwood Marsh Checker Bloom. So I was doing my research and I was like, okay, well, I guess I'm pretty much just trying to figure this one out. And luckily there's a lot of other checker blooms out there that people have grown. So there was good information there. And there was one other person that had grown it, but didn't get a lot of help from that person. And so the goal was to produce more seeds and then not put them back out into nature, but to then store them at the UC Botanical Garden with Holly Forbes. And so I actually started the seeds in my garage in a mini fridge. So they all were in little tiny 4-inch pots and required cold stratification. And so I put the seeds at the top of the pots and then put them in cold stratification. And then they were in there for about 3 weeks. And then after that, they came out to my backyard. So I've actually done quite a few endangered plant projects in my backyard. And after about 10 days, they started germinating, and I was like, thank God, like, oh my gosh, like talk about some pressure for growing plants. Like if you got the last of a species seeds in your hands, and you don't want to mess that one up. So I was very happy to see these cute little cotyledon leaves popping out of the little bean-shaped seed that the Kenwood Marsh checker bloom has. They're very cute. So the results. So it turns out that all of the seed was still viable, even though some of it had been kind of stored not in the best way. The 2007 seed, I think, before they had the equipment that they have now, wasn't stored in refrigeration. So the germination was lower. It only had 16%, but the 2008 seed, only a year later, but had been kept in the refrigerator, had 42% germination. And then the 2019 seed, which was the majority of the seed that I was working with, had 51%. And so basically I took, 82 plants and produced 33,000 seeds for this endangered plant. And then I also got to go out to the Kenwood Marsh and collect off of the wild plants that were there, and was able to collect 11,687 seeds. And then all of that went back to the UC Botanical Garden. And then I also got to just document the propagation protocol for the species, because, all of this stuff that we're doing, it needs to be well documented so that it can keep happening moving forward. So that was that one. Here's some pretty pictures of the process, and I love taking pictures. So this slideshow is very heavy on the pictures, but I also have to keep it moving. So, if anybody wants to see more of these pictures, I am happy to share the PowerPoint presentation afterwards. Okay, then after I got done doing the Kenwood Marsh project, I got the opportunity to start growing plants for the Laguna Foundation's restoration projects, and for also our conservation team. So, all this stuff that I'm telling you that, I did, really was our team that did it at the Laguna Foundation. The Kenwood Marsh checker bloom, I'll say I did that. But all the stuff I'm going to tell you moving here forward is all of our conservation team. And I'd like to take credit for this fancy hoop house where we grow the endangered plants, but Asa, who's going to talk to you, and Sarah Gordon, who's in the back, they're really the brains behind the operation. I just grow the plants. So anyways, so in 2022 and 2023, we grew Sonoma Sunshine and Burke's Goldfields in the hoop house. And then currently we're growing Sebastopol meadow foam. And all three of these species are endangered vernal pool species that really only occur mostly in Sonoma County, and a little bit in surrounding counties as well. So these are really special plants that, whose habitats has been highly altered by humans. And so we need to do what we can to help bring these things back. And so the goals of our seed amplification hoop house are to enhance and expand the population of Sonoma Sunshine at Sonoma Valley Regional Park, which we did, and it's doing very well. I'll tell you more about that. And then also to expand the populations of Burke's Goldfields and Sebastopol meadow foam at some of the other sites that where we're working. So sometimes when we grow the plants, they go right back to where they came from. And then sometimes the populations in the vernal pools are so small and we can't necessarily just collect seeds from that one site. We have to diversify on where we get the seeds from in the beginning. All right, so here's my favorite, Sonoma Sunshine. It's just like such a happy flower, and it's produced really good results for me too. So it's been easier than some of these other ones. But it's an annual herb in the Asteraceae family and the sunflower family. It grows in vernal pools, like I was saying, and there are just a handful of populations that are naturally occurring for Sonoma Sunshine left. So that is why we wanted to take this on. So same with the Kenwood Marsh checker bloom. A lot of the threats to Sonoma Sunshine are caused by us humans, loss of habitat fragmentation, climate change, a lot of thatch accumulation from invasive plants around the vernal pools ends up in the pools, and also other invasive plants that are kind of encroaching on that habitat as well. So the Sonoma Sunshine in 2022 We actually grew it in the greenhouse first in these plug trays. So I love growing plants in these little plug trays because you can be very precise. And okay, I'm going to put one seed per cell, and okay, I had this many seeds to start with, this many germinated. So it's really easy to keep track of germination success with these little plug trays. And they seem to do really well in there. And again, starting a new job, getting handed some endangered plant seeds, was very, very happy to see that these germinated, and actually 100% seed germination for the Sonoma Sunshine. So pretty good. And, not like the Kenwood Marsh Checker Bloom where we had like 50%, 17%. We got 100% on this one, probably thanks to Betty and her awesome greenhouse that I got to grow them in. And so after they germinated, we then transferred them into our hoop house. So they started in the greenhouse underneath the misting system. Then we transferred them to the shade house underneath the shade. And then once their roots were well developed enough, we put them into the hoop house, into the raised beds. They flowered about 4 months after transplanting them. And then we started collecting seeds about a month later. And then in 2023, we did Sonoma Sunshine again, but this time we skipped the greenhouse and went straight into the raised beds, and it actually worked really well. And you'll see the results of that in a sec. So here's just, some pictures of the seeds, and then the seeds starting to germinate, the cotyledons, them starting in the trays, what they looked like before they got transplanted. This is what we really want to see. This is what they started doing in April. And they were just glowing, and I was glowing, and it was very exciting. And then not long after that, about a month after, we had seed production. So this is, the seed that I was able to vacuum up in the hoop house. So this is what's amazing about this design that Asa and Sarah came up with, is that we have raised beds, and then in between the rows, there's weed cloth or weed fabric, and I can actually come through with a vacuum and just vacuum up the seeds. It's incredible. With the Kenwood Marsh Checker Bloom, it did not go like that. It was like hand collecting seeds off of each plant, trying to keep each plant separate. That was a real pain, but this, went really smoothly. And we ended up in 2022 producing 68,000 seeds off of 300. So we started with 300 seeds, we made 68,000. And then in 2023, when we direct sowed, we ended up producing 102,000 seeds. And then all of those seeds went back out into Sonoma Valley Regional Park, where the population had really been in decline. In 2018, after the Nuns fire, it was doing pretty good, but then we had 2 years or a couple years of drought. And in 2022, this is what the state of the Sonoma Sunshine population was looking like. And it was really just in one pool, and in that one pool, it wasn't doing very well. And so after we reseeded in 2022, all right, let's see. Yeah, I think it was, in 2022, we seeded in the fall. This is what it looked like in spring of 2023 with all the seeds. And so it was just in one pool, but then we reintroduced it into, was it 5 different pools? I don't know. There was many pools that didn't have Sonoma Sunshine. I mostly got to be in charge of producing the seed, and then I got to help a little bit with putting it out there, but then it goes into Sarah's hands. So then, it's like, does this continue to work? That was the question. And so in 2024, just this past spring, it was looking really good again. And so, I think that this population is not going to peter out for quite a while. So we did what we meant to do, and it seems like it's still going strong. And then Burke's Goldfields was the next endangered plant that we grew alongside Sonoma Sunshine. I'm probably not going to go through all of the details on, why it's endangered, because it's pretty much endangered for the same reasons that the Sonoma Sunshine is. So altered hydrology, habitat loss, humans. Go back. This plant has very small seeds. So when you try to sow these seeds, you may not breathe, and you need to listen to very calming music to make sure that you do not mess up. So you're like sprinkling these microscopic seeds on the top of the soil. For this one, you didn't even bury it. It was just surface sown. But you're really trying to get, a couple of plants per cell, because you don't want these things to be, over sown whenever they get transplanted out. But same thing, we started them in the greenhouse the first year, and then we transplanted them into the beds. So you can see on this picture on the bottom, the setup of the beds with the weed fabric in between the rows. And then we started having flowers and even more flowers. And so this was what it looked like when it was in full bloom. And again, I was just like blown away that we had all of these flowers. They were buzzing with pollinators, like all sorts of really cool ones, not just honeybees, but like all sorts of native bees and butterflies. And then we, once I think it was June, we started harvesting them. We did this one a little bit differently. We actually took a little grass trimmer and grass trimmed it and then put that into bins. And then we dried it in the greenhouse, because this time of year we're not really using the greenhouse in the nursery. And then after we had kind of cut the top off and had all of that seed in those bins, then we came through with the vacuum and vacuumed it up. And that's what the seed looked like in the vacuum after we had vacuumed it up. And so this one, because the seeds are so small, we got to produce 42 million seeds in the first year. And then the second year, we didn't do quite as well, but we learned a lot. And so we tried to do the same thing that we did with Sonoma Sunshine and direct sow the seeds, but it was much more challenging to direct sow little tiny seeds and then keep them right where they needed to be to germinate. And so, in the future, if we did this one again, I think we would go back to the growing it in the greenhouse into plugs. But Sonoma Sunshine, we would continue to do direct sowing. And also the next species, we did really well with the direct sowing as well. So, again, we got to produce all of these seeds. They got to go back out into vernal pools, actually a newly created vernal pool in Sonoma County from some mitigation work. So that'll be really exciting to see how well these all take in that situation. Not vernal pools that are natural, but like newly created ones. All right, so same deal with this one. We're going to kind of skip through like why this plant's endangered, because again, vernal pools in Sonoma County, same habitat, same threats to why the population is in decline. And we'll get to the stuff that I really like talking about. And this year, we did something a little different. We had some new folks come on to the conservation team. One of our conservation technicians, I'm not sure exactly what her title is, but she started taking over doing the hoop house with Asa, and then they needed somewhere to grow the meadow foam that was away from the Laguna Foundation, away from the other populations that they were growing in the hoop house, because it was a genetically different population, and they didn't want cross-pollination happening. So I got to go back to my home garden beds and convert those into seed amplification beds. And so that's what I've been doing now, is growing in my backyard again. And they've had great success in the hoop house with Sebastopol meadow foam. And before I worked at the Laguna Foundation, they'd actually grown this plant in the nursery in pots. And that's why the hoop house got created, because they realized growing these plants in pots and trying to seed collect them and keeping the seeds from going inside the soil into the pots was very challenging. So the hoop house has just made this all way more easy to do. And so in my home beds, I started the seeds in plug trays again, and didn't get 100% germination, had to do kind of 2 rounds of sowing, but every plant and seed is different. And then they were outplanted in raised beds in my backyard in 2024. We had the first flower starting in April 22nd. And now we have seed production happening now. I wish this picture was a little bit bigger. You can see all of what was flowers now turning into these little nutlets. And then, pretty soon, probably come June, we're going to be vacuuming up the seeds same way in my beds and then in the hoop house. And we're hopeful we'll get, hundreds of thousands of Sebastopol meadow foam seeds. And next year, we're going to hopefully be growing another endangered carex species. And I just I love doing this because I feel like you're kind of like pioneering something, you know, and it's like learning to grow something that nobody's ever grown before and then helping to give back to nature and really like, you know, our goal is to get these species delisted. And I think that we're on our way. We're doing pretty good. And then acknowledging that, again, this was not just me that did this. This is our whole conservation and restoration team at the Laguna Foundation, and couldn't do it without Sarah Gordon and Asa. And you're going to meet Asa soon, but Sarah Gordon's sitting back there too. And really, like, you know, she's the brains. I just do what she tells me, and you know, and we did pretty good. And then these are, you know, a list of our partners and other people I would like to thank. Again, I want to thank Betty Young because, you know, she's really like just has like inspired me in so many ways, and so it's really great to get to do this alongside her as well. And we have a video, but I don't think we have time. So I'll share it with you though. All right, thanks you guys.
As you can see, I'm going to talk about the work that the nursery has done on the Vine Hill Manzanita. As you may know, same world as the
There we go. Yours.
Okay. So many of you may know about the Vine Hill Preserve. It has been a CNPS owned property since 1976. And on the preserve, there are actually 3 endangered species. They are California listed because the feds don't have any money to list anymore. One way to save the budget for the Fish and Wildlife Service. But we have the Vine Hill Manzanita, our Distaphylos densiflora, which I'll be talking about. We also have the Vine Hill ceanothus, which is not such a happy story. And we have the Clarkia imbricata, and that brings up Sarah again. Many of you may know Sarah. She's been a member of the chapter for decades, and she was for many, or is still, the Vine Hill steward as a volunteer with CNPS, as opposed to her professional position at the Laguna Foundation. She's been working very hard on expanding the population of the Clarkia imbricata. And so that's still happening at Vine Hill. But all right, which one are we using here? Did I use that one? There we go.
Okay.
So this is Vine Hill Preserve, which many of you have probably heard about. The picture on the left is how it looked in 1976 when it had just was a vineyard. And I think this is old vines laying on the ground, and the property came up for sale. The line of plants right up at the top of the hill underneath the trees, which is right along the road, where Vine Hill Preserve were at that time the only Vine Hill Manzanitas in existence. Again, loss of habitat due to development, mostly. So CNPS understood the importance of that and did some fundraising and actually purchased that property. It's the only property in the state that CNPS owns. And then they asked, the main office in Sacramento asked Phil Van Solen to take on stewardship of the site because he's renowned as a Manzanita propagator, previous owner of Calflora Nursery. And so he started propagating from those, at that time, about 20 plants that were naturally occurring there and were somehow not pulled out when they put in vines. And so he's covered from those original naturally occurring 20 plants, he propagated about 75 or 80 more and planted them down the hill so they're not right by the remnant plants, so they can be kept separated, what were clones and what were naturally occurring. And so that really saved the species at that time because he did that propagation. Because even back then, within a few years, those naturally occurring plants were showing symptoms. And since then, things have not gotten any better. This is what on the left, what the Vine Hill Manzanita, many of them on the site, look like now. And this is the better looking of the two Vine Hill ceanothus that are on the site. And I think now we are probably down to one. We have two we propagated in the nursery. And anyway, the reason these look like that is because they are infected with the root rot waterborne pathogen that I'm always haranguing on, and that we go to great lengths to exclude from the nursery. It's your typical root rot. And so over the years, these pretty much the plants on the site have become infected. And it was so bad that in late 2019, Sarah called me and said, "Ooh, these look really bad. It kind of looks like it might be Phytophthora. Would you come and check them out?" So I did, and we have a nice, easy way to test for Phytophthora using, believe it or not, pears. They react to the spores, and you get these blemishes on the pears. I took 4 samples of soil at the site and tested those 4 samples, and you can see all 4 came up positive, which is not good news because I was in the 4 corners of the site testing, both in the naturally occurring area and down the hill where the clones were planted. So then we called in the California Department of Food and Ag plant pathology lab folks who happen to have a grant because of the new awareness about Phytophthora and how dangerous it was in natural areas. And so they sent out a team of plant pathologists who went through the site and took multiple additional samples. You can't read this, but pretty much they say every sample they took came up positive. The Milo Baker board realized at that point that we had to do something drastic to save the species, because it's only a matter of time. The spores enter through the root system and then travel up plants and they will die. So the board approved funds to hire Dr. Ted Zwicky, the renowned plant pathologist on Phytophthora in wildlands, on diseases in wildlands, and he's concentrated on, because there is so much on manzanitas, the endangered manzanitas that are being affected by Phytophthora. So he came out to the site and took samples at all these spots throughout the preserve. So the road and the remnant manzanitas are here, and then this is a slope, north-facing slope, and you can see it's traveled all the way down the hill, and the whole 1.6 acre preserve with the last remaining population of Vine Hill Manzanitas is totally infected. These plants will die these plants will die. So we needed to do something if we wanted to save the species. So that's, yeah, what much of it looks like again. So with consultation from Ted Zwicky, the plant pathologist, and from Central Office of CNPS, we decided we needed to propagate clean plants and then find several new homes for them. We can't rely on having a species only be in one place like this. If Sarah hadn't noticed and we hadn't intervened, the species would be extinct. So we needed to propagate clean plants and then find at least 3 new homes. So if something happens in one place, it is still occurring in another place. And then plant and care for those manzanitas so they're happy in their new homes until they are able to establish on their own. So we started propagating in early 21 with the help of the nursery volunteers. Nursery volunteers, raise your hands. We got a few here tonight. So we took cuttings that first year from 11 plants. Mostly I wanted to perfect, as Ayla talked about with seeds, I want to perfect the propagation techniques. So the cuttings from those 11 plants, we ended up with 33 plants. And if you're interested in propagation protocols that were most successful, we can talk about that later. Those are them. If you're a propagator, you know what that means. They were potted up the following spring, about five to 6 months usually to root manzanitas, and then into large pots by fall of 21. In the meantime, which is what's so amazing about this experience with the Vine Hill Manzanita. In 2020, Cal State East Bay now got a big grant donation from somebody because they decided that they needed a genetics lab. And so they built a genetics lab at the college, and then they went looking for projects that could help save endangered species. And they called Sarah and said, we want to test the Vine Hill Manzanitas there to see how much genetic diversity we have, especially because the plants that were planted were, of course, clones of the naturally occurring plants. And we want to make sure that's not just one big clone from a nice healthy bush, 75 identical clones. We want to see how many of the original genetically individual plants that were propagated and planted at Vine Hill. So Sarah and her father, I believe, went out and put numbers on each one of the 75 or 80 manzanitas that were at Vine Hill at that time. So each individual plant was identified. with an A and a number for the naturally occurring and a B and a number for the clones. The nursery propagators remember this because every single pot had to have its number, so we knew which exact plant it came from the preserve. And we found that, or they found, I had nothing to do with it, the genetics lab, it took almost 2 years to get results. But they found that there were 20 genetic individuals, some where we still had the naturally occurring plant and all of its clones. So you can see, you probably can't see, let me tell you. The first number here is the number of, if it's an A, it's the naturally occurring plant. So we have here in genetic group 15, we had still alive the naturally occurring plant, and then 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 clones of that planted in the preserve. So then I could go back and match up the plants I propagated with this list of genetic individuals, and found that we had only captured 3 individuals of the 20 on the site. So Natasha and I headed back to Vine Hill with lots and lots of Ziploc bags and collected plant cuttings from to capture from at least one or two plants of each genetic group. So it's extremely critical where we're down to such a small population that as we're establishing new populations, we have as much genetic diversity as possible, because with climate change, with everything else, that one genetic individual may not survive. We need as many genetic individuals with some variation so they can continue. And so we, as I said, we collected more cuttings from the field. We were lucky, did I say that? That Phytophthora begins in the roots. It invades the roots from the water in the soil and then travels up the plant. So we were able to get clean cuttings by taking tip cuttings where the disease had not yet reached. So these were additional cuttings from one plant, which we and the nursery volunteers propagated and ended up with a total of 153 plants ready to find a new home. We had, before beginning propagation, because this is a California endangered plant, we had applied for a permit. You can't just go out and willy-nilly collect endangered plants. You have to get permission from California Fish and Wildlife if it's California listed. So we did that before ever starting propagation. Let's make that very clear. You cannot just go out and take cuttings of any endangered plant. So now we have the plants. While those plants were growing in 2022, we started looking for places to plant them. They needed to go into protected preserves. Many people said they would plant them in their yards. Well, that's not saving a species. We need a population, genetically varied population of the plants in the wild, in a protected preserve where they will be able to live in perpetuity. Houses get sold and people mow them down. In fact, the reason we're down to the last couple of plants of Ceanothus is that the Ceanothus that occurred along the Joe Rodota Trail got wiped out either by the neighbor or parks maintenance crew. So you just need them in a protected area. So the sites we found, we checked, we took soil samples, of course. We weren't going to just plant these nice, wonderful, clean plants into an infected site and go through the whole thing all over again. So I took soil samples, tested those, each of the places we were going to plant. And that year we ended up planting in 2 sites thanks to Sonoma Land Trust. They allowed us to plant in the upper reach of the lower Pitkin Marsh. And we also planted in our own conservation easement at Cunningham Marsh, thanks to the landowner there, our volunteer and steward, Marcia Johnson. So we planted on her property that's within the conservation easement. Thanks to volunteers, many of them in this room. And I have pictures, lots of butt pictures. People planting. All of the propagated plants, of course, were tested multiple times to be sure they were clean, free of Phytophthora. As I said, the soil at the planting sites were the soil at the planting sites was negative. CNPS biologist in the central office wrote a planting plan, and we revised our collection permit to include planting and to get the okay to plant them at the sites that we found, where there was a historical record. Well, certainly Pitkin Marsh. I'm sure they occurred there once upon a time. The soil was the same type as is at Vine Hill Preserve. And so meanwhile, recruited volunteers and did some talks to get the word out about what was going on, and especially about the genetic testing and how important that is if you're trying to establish new populations. And Miss Dalton, Penny, and I, part of my permit is to do agrophos treatments, which is the chemical they use on trees to slow down sudden oak death, same genus of water mold. And Penny goes out every other year with me with our backpack sprayers. I'm not sure we'll be able to do that next year. And spray the 80 plants at Vine Hill to slow down the disease there. And so in January 2023, we planted 49 plants at Pitkin Marsh in the upper part, not in the marsh, thanks to Sonoma Land Trust. And I had 25 volunteers lined up, and every time we set up a planting day, it was, the atmospheric storm. So we finally, the board okayed funding, and Laguna Foundation was nice enough to pretty much charge us their cost for their crew going out and planting, along with a couple of our volunteers, including Lynette Bauer. And then there's our butts. You can recognize a lot of these people who planted in Cunningham Marsh, another 19 plants. And then this winter, we planted more plants in the Laguna Uplands, which is actually owned by Laguna Foundation, so protected in perpetuity. So we have those 3 populations established. We hope to expand them. On top of that, just to preserve all those genetic individuals, 123 plants have been taken to 8 botanic gardens, every single botanic garden along the coast from Santa Cruz to Humboldt, including just recently to Sonoma Botanical Garden, which has now decided that it would be okay to plant some natives So we're happy about that. Then taking care of the plants, we had 2 watering events last summer. Thank you, many volunteers who are in this room and not in this room. And then Lynette and Natasha and I and Judith. No, it was Judith, Natasha, and I went out in February and March and weeded all of the plants at Pitkin and monitored them. So right now, as of then, we have about 55% survival, which isn't bad because those plants got planted and they were on their own. Some of them look better than others. The one on the left is big and blooming in March, and one on the right, not so much. And Jan Lochner and her team, and Marcia in particular, have been weeding the Cunningham Marsh Manzanitas, and Marcia has been watering them. And so it takes a village. And I think all of the people out here who have helped us with this project, we also collected seed and are playing with part of those, and part of those went to the same storage facility at Berkeley that Ayla has taken those seeds to. So thank you. That's fine. Oh, introducing. Give me the mic. So Asa, you're up. So Asa started working at Laguna Foundation in 2015, almost 10 years ago.
Oh my.
And
That one's not on.
no,
Sorry, buddy.
Oh yeah. I got to know Asa because he is a wonder with equipment raised on a farm. And you can't underestimate the value of somebody who was raised on a farm for fixing equipment. In fact, the tractor right now, the Laguna Foundation's tractor is sitting at Cunningham Marsh because it broke while he was weeding, and he's got parts on order and we'll fix it. Yeah. And finish the weeding. Because he did such amazing work with the Laguna Foundation, he was promoted and is working with Sarah Gordon. She is, if you don't know, Laguna Foundation's vernal pool and wetland biologist. And so Asa works with her on vernal pools, and because Cunningham is a wetland, when either Sarah or we wrote grants to do work at Cunningham, most of the heavy work, then we went to Laguna Foundation because the crew is so amazing and dedicated and careful. You could trust them when the leaves of the lilies are only this big, not to mash them. So Asa has really become the manager for the Cunningham Marsh, and we continue. The board has been allocating funds to just hire Laguna Foundation each year to continue that on. Asa also has his fire burn, he can burn. So I will turn it over to Asa to top it on.
Thank you, Betty. Okay, good evening, everyone. This evening, I'll be presenting on the Pitkin marsh lily, the Lilium pardalinum subspecies pitkinense. But before I break into that, I do want to just kind of give a shout out to my organization and our program, and kind of give you an idea of what we do. So I am the preserve manager for the Laguna de Santa Rosa Foundation Conservation Program, and our focus is on rare and endangered plant restoration. And we work on a number of endangered plants in the Laguna de Santa Rosa watershed, and also a couple that are just outside the watershed, because we like to bend the rules a little bit. A lot of our work focuses on vernal pools, but we also work on 2 wet marshes and 1 disjunct chaparral habitat, actually, Vine Hill, which you saw earlier in Betty's presentation there. And there is some of the beautiful endangered plants that we'll be working on. And of course, center stage, I had to put the Pitkin marsh lily. A lot of our work is focused on managed beneficial disturbance. Many of our endangered plants are not doing well, partially because they're not getting the natural disturbance regimes that they have evolved to thrive in. And that may include cultural fire use or natural herds of ungulates. So we are working to put animals, mowers, and fire back on the ground so that we can reestablish healthy endangered plant populations. Maybe one day getting them out of being endangered, but let's keep our fingers crossed there. One big component, which I'm not going to go into too much detail because Ayla described it earlier, is our endangered plant seed amplification hoop house. And you can see Burke's Goldfield, and this is our current run of the Sebastopol meadow foam on that photo on the right. And the Laguna Foundation only owns 1 piece of property, but all the properties that we work on are from various landowners, public agencies, private landowners. And so, and like Betty was saying earlier, when you hear about endangered plants, you're thinking about some exotic plant that's located in the Amazon. You're not thinking about the ones that are literally in the middle of these suburbs and on the roadsides of 101. But here in Sonoma County, we have these very unique specialized habitats too. And we think it's very important that we connect people with them so that they show that appreciation and can protect those places into the future, because many of those species are endangered because of habitat conversion to residential development or agricultural use. So a lot of our program is about getting involved with the community and bringing up their awareness. And then in any successful habitat restoration projects, you need to build a close relationship with the habitat and the species you are managing for, but that's especially true with your endangered plant species. There's inherently not many out there, so the risks are much higher. There's also not the data or knowledge set that's out there. So we have to spend a lot of time getting to know the plant before we do anything that we think can better that plant. And that's through annual monitoring and surveying, extensive field notes, and just really getting up close and getting to know that plant. Numerous site visits to see it throughout the different seasonalities. If it's located in multiple sites, we have to go visit those multiple sites to see really the range of what's available for those species. And that's definitely true for the Pitkin marsh lily too. And really, honestly, this is my favorite species that I get to work with. And anytime I'm working with it, like I really do have a smile on my face. I think this plant is gorgeous, and I'm in love with its habitat. Okay. So the Pitkin marsh lily is historically documented from a confirmed 2 locations. The Cunningham Marsh Preserve, where it's still at, and the Pitkin Marsh Preserve, where its namesake. The population at the Pitkin Marsh was actually not in the preserve. I'm sorry, it's in a neighboring private property, and it has not been observed in a number of years, and it's assumed to be extirpated from that location. So for all intensive purposes, We only have one confirmed location remaining in the wild, and that's at Cunningham Marsh. It is also, it's a subspecies of the leopard lily, and is one of the 5 subspecies of the leopard lily. It's a clonal herbaceous perennial plant, and it's native to wet forests and marshes. The plant takes several years after establishment to become a productive individual where it can flower and produce seed. From my observations, that's about 3 to 5 years in the wild at that Cunningham location. So it really does take a while and an investment from getting that plant to seedling to being a fruiting, productive member of its society. So almost all occurrences of the lily at Cunningham Marsh are located near and under the canopy of black oak trees, where the shade and leaf litter provides some competitive benefits against invading herbaceous species, mainly the purple velvet grass. So it likely, historically would venture out from the shade of these black oak trees, but currently we see the black oak as a critical piece of its habitat. And a lot of our work is focused on preserving our heritage black oaks and reestablishing those black oaks so that we can have good productive lilies. Endangered plants, there's not much known about the pollinators for this species, but the greater leopard lily is known to have been pollinated by hummingbirds, butterflies, native bees, moths, pretty much all the suite of native pollinators. And that lily you see in the center with the little native bee, that's actually a leopard lily from the Lassen area. I've actually never observed a pollinator on the Pitkin marsh lily. I know it's occurring because every year that the productive individuals produce copious amounts of seed. So we know it's occurring, but that information we don't have. Mature plants emerge usually around late January through mid-February, with the germinates occurring just following that. So we're talking about early February and stretching into mid-March. And the germinates, I mean, they regularly germinate. We get tons of germinates, hundreds, if not thousands of germinates each year are observed. The problem really with that is establishment. Establishment rates for this species are very low. And if you see here the photo on the left, you have a whole entanglement. I don't know if you can see the little germinate leaves within the black oak leaf litter, and showing their roots kind of growing in and amongst that litter, but seemingly having low establishment rates due to the poor seed soil contact. And you can actually see this one over here in my hand. This little germinate is forming a bulb. I just want to point this out because I think this is so cool. You can see the seed husk right here and a bulblet already exists, and that was just growing on top of a black oak leaf there. And so when you run across these ones when you're doing your hand weeding, you have to poke a little hole, get it back down into the soil. Just walking back in this really quick, we've seen improved establishment where there's areas of disturbance, where the lily reaches over its exclosures and drops seed on deer trails is one area that I've seen them be successful. So we really do think that managing that mulch and leaf litter is going to be essential to getting good establishment. Bloom occurs between mid-June and July, early July. And when it blooms, it is one of the most beautiful flowers out there. So that's why it's my favorite. And this photo here is showing peak bloom for the species in its most productive, and in one of its most productive patches. And the species generally enters dormancy by late September to mid-October. And you can see those seeds hanging out in those dried up capsules there. And seed distribution is generally limited to basically the length and lean of the floral stalk. You don't see germinates occurring more than 2 or 3 feet past the productive individual. I haven't seen any wildlife utilizing the seeds or spreading them that way. That may be because they're currently in exclosures, but it really is limited to just how far that stalk leans over and those seeds drop. A bunch. I've never counted, but like a bunch. You know, I'm gonna guess between 20 and 70. That's a good question. Now I got to count. Got more questions. And the species is readily browsed upon by wildlife. Rabbits and deer are the most common and most well-known species that browse upon it. It's possible that rodents are utilizing the seeds for some kind of food, but I have not seen that. But this like preferential browsing by wildlife is what necessitates us building and maintaining these exclosures around them. So everywhere where there is this Pitkin marsh lily growing, we have built cages or fencing around them to keep wildlife out and protect those plants from herbivory. And it's really important too, because as the wildlife browses on these plants, it pretty much means that plant that's been browsed upon will not produce seed or fruit that year, rather. And for a species that is so limited, we can't spare to lose a single year of productiveness. So right now, a lot of work is focused on maintaining those exclosures. So there's no ethnobotanical history known for this species, no data about human use for the species, but the greater leopard lily is well known for its cultural use, and it's very likely that makes up a big piece of the natural ecology of this species as well. And some of the stewardship tasks that, rather I'll walk it back a little bit, we were brought on in 2019, funded by a United States Fish and Wildlife Service grant from their partner program to reduce the invasive pressure on the exclosures in the lily habitat so that the CNPS volunteers can more easily manage those lilies, because they've been managed through careful hand weeding around those little plants to keep them healthy and happy. And this photo here on the right, Jan Lochner actually sent me that photo, and she had a caption, "Beauty and the Beast." So you can see the beautiful Pitkin marsh lily growing intermixed with the purple velvet grass. And unlike this Beauty and Beast, there is no, romance or love story here. We also implement different forms of vegetation management. As I described earlier, one of the things that we believe is inhibiting recruitment or establishment of these seedlings is the overly thick black oak leaf litter and forest mulch or duck. And so what we're doing is we're bringing in brush cutters, and we are cutting any of the woody vegetation like poison oak or Himalayan blackberry that competes with the lily. We're also working to kind of mechanically grind up or break down, decompose that leaf litter, and work those seeds deeper into that soil profile so that we can hopefully get better establishment. Since this species takes several years to produce a productive individual, and it's currently being surveyed by capsule counts, or counting the finished up flower, none of our work yet is being shown within that data set But anecdotally, visually, with visual observations, we have a dramatic increase in young lily plants. And so I think we'll be seeing those numbers rise in the next couple of years. And that's really important too, because as we have more productive individuals, they start producing more seed and gives us the opportunity to really expand those populations and make them more resilient. So the Western azalea is known to be a productive habitat component for the Pitkin marsh lily. It's thought that the azalea protects the lily in the wild from deer browse as the lily kind of grows intermixed with those woody branches there. It kind of limits the access that deer have to it. And that's definitely true. But as these azaleas become 6 to 8 to 10 feet tall, and they have many clonal sprouts growing at the base, it becomes where they start competing with one another. Rather, the lily is not competing with the azalea. The azalea is winning the fight here. So some of our work has also been performing experimental pruning and thinning of the clonal resprouts. When we thin the clonal resprouts, we'll plant them out in other locations in the marsh to improve and expand that suitable habitat. And anecdotally, we've seen a great uptick in productivity of the individuals growing around the azaleas. A lot more blooms, a lot more individuals, and greater ground cover with the lilies. So far, this has been a really effective experiment. We're only working on a small scale because when you're limited to a single population, you don't want to go big. Start with one, pruning one azalea and look at the effects, and then, okay, next year, what if we prune it like this? We're still in those stages, but it's looking very promising. And our work here at Cunningham Marsh is also very much focused on native plant stewardship. The marsh, when we started there, was pretty much, it's fair to say, covered in impenetrable blackberry. And I know many of you volunteers are familiar because you've been working on it. And the question was like, what's gonna happen when we pull that blackberry away? Are we going to see a native plant response, a resurgence from plants emerging out of the native seed bank? And we've been delighted to see that yes, there is a native seed bank still present there, despite how long that area has been covered in blackberry. And different tasks for stewarding those native plants is just, it can be watering them, hand weeding around them. This picture on the right is showing an exclosure, which was formerly completely covered and surrounded by velvet grass. We had put tarps down, and we kept those tarps on there for a year, long enough to kill all of those plants. We pulled the tarps back, and then my team with CNPS volunteers, we collected field divisions from sedge and rush that was collected on site, and planted it surrounding the exclosure. We've seen good recruitment of the lily occur in the rushes, especially when there's deer trailing through it too. It seems like they kind of like punch and incorporate those seeds and make a nice little pocket for them to get established. And this is really like so fun because it's a native plant treasure hunt. And you can see Jan there on the left, cutting away at blackberry that's really impenetrable and well over her head. And what she's looking for is not only to remove that blackberry, but to find those remnant native plants. And when we find them, we flag them and we protect them, like this little coffee berry here on the right. But we also find Western azalea, ribes, hawthorn, all kinds of really interesting, cool native species. And when we're cutting this blackberry out, my crew's doing it with like mechanically, with like brush cutters with metal blades that kind of grind up and cut down the blackberry. It's a form of beneficial disturbance that we're introducing it back into this habitat. The photo on the left is 1 year after blackberry removal, and you can start seeing the native sedges occurring. Those aren't planted. That was absolutely pure blackberry. The photo here on the right is that same area last week, and now it's completely covered with native sedges and rushes. I actually looked at that photo, and everyone's shouting out Sarah, so I'm just gonna shout out Sarah too. Hi, Sarah. But I was telling Sarah, I'm like, we might have to do something about that rush. It's coming a little bit thick. Whoops. But like finding and like supporting this natural regeneration is so fun. Like I said, it's a native plant treasure hunt. But even more exciting is the less common species. So on the left, that's Asa Voigt natalii. It's a fern ally. It's a wetland obligate, and it was growing. I just observed this last week growing in the exclosure with lilies. Even more exciting than that, this species on the right, witch's teeth, is a Hosakia species. That's a rare plant, and we have not seen it at Cunningham in all of our work. This is within an exclosure, seemingly emerging from the seed bank. So it's kind of been on my bucket list to, find a rare plant in the seed bank, and I can finally cross it off. I've been looking for the Cunningham Marsh cinquefoil, and I still got my fingers crossed, but I'll take Hosakia for now. This is really exciting, and that's a perennial herbaceous plant. I'll be keeping an eye on it from now on. Very exciting to me. And earlier I talked about how we are kind of viewing the black oaks as critical habitat for our Pitkin marsh lily. They are giving the competitive edge. In areas where the black oaks have fallen, where there has been like productive Pitkin lily patches supported by black oaks, and those black oaks have fallen, we see those areas being readily and rapidly colonized by velvet grass. And it's those herbaceous invasive species that are really the chief contributor to reducing habitat quality for the Pitkin marsh lily. So it all starts with managing our black oaks, and black oaks are susceptible to Phytophthora. So we introduce sterilization protocols at the marsh just to ensure that we're not tracking in any pathogens that would jeopardize any of our oaks and thereby jeopardize our lilies. This photo here on the left is the oldest black oak I've ever seen. It's the oldest at the marsh. It's about 6 feet wide in diameter and probably well over 400 years old. It's an incredible individual. And our oaks are heritage black oaks. They're kind of aging out. Every year we get a few that topple over, including ones that formerly supported Pitkin marsh lily habitat. So it's really of the utmost importance to protect the individuals that we have left. You can see someone climbing here, this little orange shirt in the oak on the right, and I have a little bit of a story about that later. And you can see this giant crack in the center photo of that oak, also part of that story. And in addition to preserving the heritage black oaks, we also have to support that next generation of oaks. And since this habitat has been completely overtopped by blackberry for so long, it has inhibited natural recruitment of oaks. Those acorns are falling, but there's no way they can get established and grow through that blackberry. So once we removed it, we started seeing a lot of new oak saplings occur. And when we see them, we flag them, because when we come through with our brush cutters, we want to protect them. That's one way that we steward those native plants. But in addition, because we're so focused on getting new black oak cover in as quick as possible, we're harvesting acorns in the fall, and we're direct sowing them at the marsh. And of course, we're harvesting from that location. Everything we're doing is with seed sourced from Cunningham or propagated sourced from Cunningham. There's nothing being brought in from outside the marsh. We're really focusing on preserving the natural genetics and biodiversity of that site. Some challenges. Of course, all endangered plant restoration comes with challenges, and Cunningham has definitely got its fair share of challenges. And this is a forested marsh habitat that's been invaded by blackberry. Also, planted Monterey pines are abundant there. Many deadfall trees. It is a fuel bomb. It's wildland fire fuel bomb. And the concern is not only for those big campaign, tens of thousands of acre mega wildfires that we're all super scared of. But this preserve exists literally in people's backyards, and the definition of the wildland urban interface. And there's tractors and mowing that's occurring. It would really only take one spark from an ill-timed mow during the summer for that fire to quickly take over and be catastrophic for that preserve and that species. So this is definitely something that I worry about every dry summer windy events, and I'm thinking about this location. Another big challenge at the marsh is just the incursion of the non-native grasslands. Cunningham Marsh is pretty much two different habitat types: a wetland forested marsh and a non-native grassland. And when our work started, much of the interior marsh where the lilies were growing had been highly invaded by the velvet grass. It was coming in on wildlife, on people's shoes as we go in to do our maintenance of the marsh. So we focus on just trying to manage those non-native grasslands so that they don't produce that seed and it's not tracked in. We also reduce the number of entrances into the forest so that we're kind of like bottlenecking that invasion to certain locations. We chose those locations where they're kind of like armored with either competitive native vegetation, or we cut it right through the center of a non-native blackberry patch, so that we can always pull the ripcord and let blackberry take over if focus becomes a major issue there. But timed mowing helps prevent the velvet grass from producing seed. It's critical that we manage our grasslands so that we can reduce that invasive pressure on the lilies. Then, of course, I was saying, the preservation of the critical black oak habitat. Every year, in every winter storm, we're losing these heritage black oaks, and we're not replacing them nearly fast enough as the rate we're losing them. This photo on the left is a black oak that not only fell, that was supporting Pitkin marsh lily, it fell on top of an exclosure containing Pitkin marsh lily. Yeah, and it's kind of a tragic situation that way. But we got a lot of successes too. Mainly increasing that native cover. Still really stoked about that Hosacea. Here is, hi Betty, I see you on the left there. There's an exclosure containing the Pitkin marsh lily. This is in 2019. And where Betty is standing, that's all the invasive velvet grass. And that was the pressure on that exclosure. This is actually a before and after photo. You can see the the exclosure kind of hiding in the background of this photo here. And now is where we see all those native sedges and rushes, which were planted by the CNPS volunteers and the Laguna Foundation crew, and is creating that competitive edge and suitable habitat so that the lily isn't under constant and direct invasion. Here's another exclosure in 2019 on the left. Velvet grass growing right up to it. And this is actually the exclosure that black oak tree fell on top of. And when it fell, it sheared off most of its roots, but there was still a little bit connected. And so, you know, careful what you ask for. I asked for, you know, increased black oak cover for my lilies, and look what happened. So I actually worked to kind of preserve this tree, and I pruned back the canopy to reduce the transpiration, because less roots sucking up water, I have to get less leaves pumping water out. And through that strategic pruning and kind of choosing where the regrowth will occur, we've actually now gotten some black oak cover very quick for this exclosure. We rebuilt the exclosure around it. This population is still healing from that event, but our native cover is greatly increasing. This is the exclosure that actually has that Hosackia in it as well. I mean, in this photo, you could actually see this is a fallen oak, and this is, in this area alone, there's about 5 or 6 of those large heritage oaks that fell, and that's where it just completely was converted to the velvet grass. Okay, this is where I'm going to bring up that story from earlier. This these 2 pictures you're looking at, this is the black oak that is supporting the most productive population of the Pitkin marsh lily. And on one of my visits there, and this is you can see that it's basically 2 large leaders, or, you know, trunks of the tree. And the one on the right starts splitting away from the main trunk on the left, creating this very large crack that was over 1 inch wide. And it was very much on its way to losing a large portion of its canopy cover, but also, if we let this continue, would have left a giant wound on this tree, 2 feet wide and 6 feet tall, and greatly reduced the lifespan and productivity of that individual tree, and really would have jeopardized the well-being of that most productive population. This is where I can give a shout out to Merlin from Merlin's Arbors Group, who actually happens to be in the back. Yes, a big round of applause for Merlin. He actually got the emergency call, I think, from Betty, saying, like, this is, we have this situation, and his crew came out and pro bono. They pruned to remove weight from that trunk on the right, and they put a cable in the tree to support it. And now, I mean, this is, you can't really see it here, but this crack is absolutely sealed. This was from a couple weeks ago. It's a healthy tree. There's slack in the cable now. It's sealed itself up. The tree is healthy, and the lifespan is greatly increased due to his and his crew's generosity there. So again, thank you, Merlin. You really have saved those Pitkins for us. Here's some other successes. It's just the photo on the left showing an oak sapling, but the photo on the right showing plantings that the CNPS volunteers planted, I don't know how long ago, maybe like 15 years or so. Those
2005.
Yeah, and they're actually starting to create canopy cover in that non-native grassland, which will help reestablish that as a forested habitat, reduce that invasive pressure. And I was just, honestly, as I was tractor mowing last week, I took this photo. I thought it was so cool. That house in the background, most people are familiar seeing it yellow. They just painted it gray, so it's a whole. They went completely opposite direction. And you know, next steps for this preserve, we've gained a lot of ground. We've made a lot of progress, establishing productive habitat again, reducing that invasive pressure, but we still have those large accumulations of fuels. We're focusing on increasing our partnerships with landowners, with other organizations, so that we can find funding that can provide for the future work at this location, and reduce those fire fuels would be number one. So that's a major concern. We have a whole list of things that we could do, but pile burning and fuel reduction work. Also prescribed grazing. Historically, the site was grazed by cattle, and that disturbance regime may have helped to preserve that species. With, you know, really our tractor mowing is kind of a Band-Aid for managing the grasslands. What we really need to do is get animals out there so that we can more heavily impact them, those non-native grasses, and at the right time of year, so that we can reestablish native cover. We can also use these animals in the forest to reduce the blackberry and other invasive or, you know, over luxuriant growth of these species, and many, many more dreams establishing this species at other locations, so that we're not at risk of, you know, losing it to, you know, one disastrous event. It's definitely super important for protecting this species. And I just want to end this with like really a, you know, thank you and a shout out to all of our partners. CNPS has been stewarding this preserve since the 80s. You all are the reason that that species still exists there. That Cunningham became a preserve. Betty, you've always been, you know, forefront on pushing these important things to the front. And Jan and her intrepid weeding group has allowed that partnership, working with Jan and her group, has allowed us to go so much further than what that grant funding allowed. We were able to come in and do kind of the heavy mechanical work, and then CNPS volunteers followed up and really focus on that native plant stewardship so we can get that native vegetation back in place. And of course, Marcia, who's a property owner, the preserve steward at Cunningham, and a Laguna CNPS volunteer. CVFW owns the conservation easement for the Cunningham Marsh, and the original grant funding came from the U.S. Fish and Wildlife Service. And we've had great partnerships with these two landowners, John Chilton and Goatlandia, who owns that property now. Without them and their support for this project, it would be much harder to move these efforts forward. So really just thank you to everyone who's had a part in this work on the species. So that's it.